Good morning, everybody. We will continue to discuss regeneration today. So we use the same set of slides. I will first briefly review what we learned because I thought that I missed some important content in the previous lecture. So we have discussed a few invertebrate, as shown on this slide. We have a sponge, hydra, flatworm, and an earthworm. So this species can regenerate themselves completely. So basically, they're immortal. They will never die. So one thing I emphasize is your appreciation of the evolutionary relation between these species, among these species. So you can appreciate how regeneration or stem cells is evolved over time. So this is a phylogenetic tree. We first described sponges. As you can see there, sponge is perhaps the first among the first multicellular species. So before it, all the species are monocellular, such as bacteria. So when you reach sponge, now this is a multiple cellular species. So we call it metazoan, right? The term for it is metazoan. It's now multiple cellular, but it is extremely simple. I will show that in the next slide. And then move on when the species evolve, and you can have something like a hydra. The hydra is now called a eumetazoan. So that means they are not as simple as sponge. They start to be developed from a germ layer, from germ layers. You start to describe them with terms like endoderm, ectoderm, and mesoderm. Although hydra only have 2 germ layers, but it is a species that start to have germ layers. Sponge, in the development of some sponge, there is no involvement of germ layers. But in hydra, there is involvement of germ layers there. So if you still recall, this is the process we described, how stem cells are involved in the regeneration of sponge. But I want you to look at the structure. This is a section of a sponge. As you can see, they don't have a lot of things, a lot of structure for you to describe. There are some water channels, that's all. You see water channels there. Basically, the water will carry food, nutrient into these channels, and then by diffusion, the cells will get their nutrients. You don't see mouth or tail or anything like that. So this is a extremely simple metazoan. So people say this is, look at there, this is the earliest extant multicellular animals, one of the earliest extant multicellular animals. So they're still, we can still observe them. They're not extincted. They're still somewhere in water. But it is very, very simple. So you start to have, even for this simple metazoan, you have stem cells involved in their regeneration. So the stem cells here are considered to be the earliest stem cell in terms of evolution. And then move on, we describe the regeneration in hydra. The hydra start to have germ layers. The 2 germ layers, ectoderm and endoderm, give rise to the 2 cell layers here. We still call them endoderm and ectoderm because they only have 2 types of cells and nothing more than that. I explained that the stem cells are located in between the somatic cells here. So the regeneration is quite simple. Basically, the stem cells will just proliferate and divide, and then because they're located in between the somatic cells, their proliferation and the differentiation will push the somatic cells to both ends, toward the head and toward the foot. Therefore, you can imagine that there is a constant cell replacement. So the old cell, the somatic cell, will get removed from the end, either from the tip, from the head region, or it get lost from the foot region. So the cell replacement is occurring all the time. Therefore, when you amputate this animal, you can imagine they can regenerate because it's going on all the time. The stem cell will push the somatic cells toward both ends. Therefore, if you cut them, of course, they will regenerate themselves. So although stem cells are involved here, the stem cells may not have a positional memory. So you see a structure. One part is called hypostome, while the other is the foot region. But I wouldn't think the head region and the foot region are very different, because as you can appreciate, they have only 2 layers of cells, and then in the middle is the gastric region. It's the stomach there, and other than that, they're very simple. The head cells and the foot cells are quite the same cells. So I imagine if you provide an anchor or provide a surface for the head region, maybe the head region now become foot. They will get anchored and then become foot, and maybe you start to see an opening from the foot region. If you remove the foot, make the foot region now become floating, right? Not attached and now floating. Probably they will generate an opening, a mouse-like structure there. Therefore, although this is a powerful creature that can regenerate themselves, but we may not be able to learn the positional effect of either the stem cells or some other mechanisms that are involved. So I don't know if you can appreciate what I'm trying to say. What I try to get this through is, although in this creature you see head and see foot, but the head and the foot cells are not distinguished, not distinctive. Probably they are convertible because they're the same cell, maybe based on where they are, whether they're anchored to a surface or not. Maybe the head region and the foot region can swap. Therefore, there's no true identity for head and for the foot. So for the cells to regenerate, they don't have to know where to regenerate head, where to regenerate foot, right? It's not necessary for the regeneration purpose. Why I emphasize that difference? It is because today we're going to describe this planarian or flatworm. So let's go back to that phylogenetic tree so that you can appreciate the relation. Then now this is planarian. The planarian is still invertebrate. It's not like they're vertebrated. They're still invertebrate, but they are different from Hydra, at least by their symmetry. The Hydra is radial symmetry. I explained to you what is radial symmetry. Basically, if you look from the top of the Hydra and then you cut the Hydra like cutting a pizza, then you will see the resulting part are almost identical to each other. That is called radial symmetry. But for planarian, now they are left and right symmetry. That's why they are called bilaterian. Bilaterian are all left and right symmetry, bear slab. So they are invertebrate, but now more advanced. Not only their symmetry are more advanced, but also the Hydra doesn't have the complicated structure like this flatworm. So the flatworm will develop from 3 germ layers, and then they have a neuron system, they have a muscle. They are just a lot more complicated, a lot more advanced than what we described, the earlier species we have described. So this can regenerate itself, and the power here is kind of amazing. I explained this from the beginning of the last lecture. I want to say this again. So you have a individual here that's a flatworm. You can cut twice, resulting in the head, the body, and the tail. And then each of these three pieces will be able to regenerate and then give rise to a new individual. So as the result, now you have three individual flatworms here. So this is kind of on side. I don't know whether you're impressed or not, but this only happens in movies, in sci-fi movies, right? You cut someone's head and then they will grow it back. This is a very amazing when you compare this to Hydra, because now this creature have eyes, have tail, have all the sophisticated structure. You will wonder, when they regenerated, how do they know, to grow a head instead of a tail on that particular dimension? So that's something truly amazing. You have this body here. How could it,
Professor?
Yes.
Sorry interrupt, but I have a quick question. So so if they have their middle part, so they can grow their head and tail.
Yes.
So so even though their head are gone or tails are gone, so
Yeah.
as much as they have their middle part, they will be able to grow their head.
Yeah, as shown here, can you see my
Mhm.
mouse? So you can
Yeah.
see that the individual get to cut to three pieces. The middle part, as shown here, will regrow the head and also regrow the tail.
Is
Therefore, that middle part become a new individual.
yeah.
What that head part, that also a result of that cut, right? But
Mm-hmm.
the head is going to regrow the body and the tail. And
Nice.
as shown here, the tail will regrow the body and the head.
So we can say that as much as they have any part of their body, either head, middle part, or the tail, they can grow the whole thing, right?
Yes,
Yeah,
almost,
it's kind
almost.
of surprising. Yeah.
Yeah, it's surprising. I told you probably before I teach this class, I didn't even know there is such creature in existence.
Yeah, it's really awesome.
Yeah, if it is a plant-like animal like Hydra, that's kind of very primitive. So basically, if the cell will grow, they are going to regenerate. But here it's very different. There's a sophisticated structure involved in there. How can the middle part will know, to which end to regenerate the head? Why there should be a head there, right? It's kind of amazing. But there are some exceptions, like if you cut the tip of the head, that's the very end of the head. If you cut right there, you leave just the tip of the head, and then the animal cannot regenerate itself. And also, the same thing happens to the pharynx. The pharynx is the throat for this animal. So if you just take the pharynx, and then that part cannot regenerate the rest. On the next slide, I will explain to you why. But otherwise, you cut this animal, in most cases, they will be able to regenerate themselves. So I guess the amazing part is how this animal can memorize the position of the body structure. That's something people are mostly interested in. So on this slide, I introduced a new concept or a term called neoblast, shown in the black font. Is neoblast is, is the stem cell in this creature. So here in the figure, I will explain to you how the new blasts were identified by experiment. So basically, you can use radiation. The gamma radiation. As long as you have a irradiation source, you can give this some dose of irradiation based on your experiment design. So you can have a low dose or high dose of irradiation. If the dose is low, for example, like shown here, it's 1,700 rate of dose, and this dose is known to kill all the new blast in this creature except the last one. So basically, you will kill all the stem cells in this creature, but you leave just one there. And of course, in some individuals, probably that dose will kill all. In some other doses, in some other individuals, that same dose may leave 2 stem cells left, right? So it's experiment. It won't be exactly just one. But in some of the individuals, this dose will leave just one. That's experiment. I would say maybe in most of the individuals with this dose, you will get just one cell left, one stem cell left in this individual. So at the same time, maybe all the other cells get killed as well. The somatic cells will get killed. And then if you give the nutrient, the culture, the correct culture condition, this single cell is going to regenerate the whole animal. It's going to regenerate the head, the tail, and all the muscles, all the neuron system. That a single cell will be able to regenerate everything. Okay, so that's one side of the experiment. And then in another experiment, the scientists used an extremely high dose of radiation. That's 6,000 rate. And in this case, it will destroy all the cells in this animal. So the dose is quite high. It's going to destroy all the cells, including the last new blast. And then from another animal, they're going to take a new blast. It's a one stem cell. It's the same flatworm, but it's a different individual. They take one cell, they put into the body of this animal, and now you can revive this animal. Basically, that cell from another individual can repopulate the body and then make a new individual. So that's the concept of new blast. A new blast is clonogenic or super pluripotent. A single cell will be able to regenerate the whole body of this flatworm. So I guess, like I said at the very beginning, the most interesting part is how the animal will recognize the position of different structure. So they know, in the head region, they're going to make head cells, like the eyes. And in the tail region, they may make body structure like epithelium and muscle. So how will they know that? So I won't explain that because it is not completely understood, but just to give you a theory. And of course that theory is based on some evidence, but as I said, it's not entirely understood. I just offer you a kind of theory. So in this theory, scientists describe that in the head region there will be some diffusible factor, such as a growth factor, like a FGF. I'll just give you an example. It's not exactly FGF. It's just a growth factor that have a higher concentration in the head region, and then towards the tail, that the same diffusible factor will get less and less. So there's a gradient there throughout the body. And in the tail region, you have other growth factors that have high concentration in the tail region. And then that same factor gets less and less when you move towards the head region. Therefore, wherever you put a stem cell in there, the stem cell will get induced by the diffusible factors. Then because of that, they know, in the head region, they're going to differentiate towards the cells in the head, such as neurons in the eyes. And they also know how, if they reach the tail region, now make more muscles and epithelium cells. Of course, that's an overly simplified explanation of how the cell will know the positional effect. But in this theory, as you can see, the stem cell itself doesn't determine the differentiation outcome. It's the environment that gives the signal that triggers the cells toward different fate. It is one theory here. Okay. Okay. Although in this university, I don't know if anybody works with this flatworm, but I know elsewhere this is quickly becoming a very important model system to study regeneration. One of the reasons is not only this species is super regenerative, it is this is probably the only creature that have the total power of regeneration, but also have germ layers. It has the neurons, eyes, muscles. Basically, this is a creature that is the closest to human that have total power of regeneration. That's why this have draw a lot of attention, and this has become a very useful model system for studying regeneration. The other reason is because there are closely related species to planaria, very, very closely related, but the other species don't have regeneration power as powerful as in planaria. So when you compare those species, you start to learn the mechanisms why this creature have the power while others don't have, because their gene expression are quite similar. They're closely related. Therefore, their gene expression are quite similar. Now you can appreciate the difference, right? If you compare this to humans, then we have thousands of genes are expressed differently. It's going to be very difficult to find what's the true mechanism there that drive the cells for their powerful regeneration. So we explained from several species, 3 species, the regeneration process that involves the usage of stem cells. In each and every case, in sponge, in hydra, and in flatworm, you need stem cells, the presence of pluripotent stem cells for the regeneration to occur. And from this point on, I'm going to describe animals that regenerate, but that regeneration doesn't involve stem cells. So stem cells are not important, they don't play a role in the process here. So,
Professor, I have a quick question when you get a chance.
Sure.
So when you perform amputation, when you amputate the head or amputate the tail, how does stem cells know where to regenerate and how to regenerate? So how do they know they should form head or form tail?
Okay. So first of all, in that flatworm, there are a lot of stem cells in there. About 20%, 30% of all the cells are stem cells. So it's crazy. It's basically a lot of stem cells are always located throughout the body of that creature. And now, if you amputate the animal, for example, close to the head region, and now along that cut, that stump, you will have some signaling molecule, for example, growth factors located there that have a higher concentration. And that growth factor can induce the stem cells towards the fate of neurons, eyes, that type of cell fate. It's because of the local growth factor concentration. The stem cell there now knows to regenerate the head cells, not the muscle cells, epithelial, that make the tail to be defined. It's because of the local growth factor concentration. It's not because of the stem cells, but because of the signal around the head region. But like I said, it is an overly simplified explanation. In reality, it's likely more complicated. Okay, so I'll move on to the second part of today's lecture, and this regeneration processes don't involve stem cells. But first of all, I would like to explain the range, scale, or power of regeneration in limb amputation. So in this part, we only discuss about limb regeneration, because in more advanced creatures like xenopods, like us, amphibians, rodents, deer, humans, all these, tetrapods with 4 limbs, right? The most studied is limb regeneration. So we learned a lot from limb regeneration, much less from other organs or other structures. So this part will focus on limb regeneration. On this slide, I will describe the scale or power in different species in terms of limb regeneration. So as you can see, this arrow here, that means regeneration power or range from low to high. The low is defined as the lowest will be just wound healing with a scar. Like in humans, if you have, unfortunately, an accident, the limb have to be removed, and then eventually it's going to be wound healing with a scar, and nothing more than that. It's just a scar to replace the limb. And then a better outcome, the better power of regeneration is scar-free wound healing. So in some other creatures, you don't see scar, you don't have connective tissue. So that wound is repaired completely, but it's not like you're going to regain the structure and the function of the limb. It's just that stump is repaired close to perfection. So you don't have a connective tissue, a big scar there. Okay, that's wound healing, scar-free wound healing is the next better outcome. And then next, along the range of regeneration, power of regeneration, you see hypomorphic regeneration. Hypomorphic regeneration means, okay, now probably you will grow some part of the structure, but the structure is not comparable to the perfect case. If you do amputation one arm, now something grew out. Okay, there is something there, but the structure is not nearly comparable, to the symmetric part. You're like a left and right. Now the right now regrew something out, but not as good. Not as good, either morphologically or functionally. It's not as good. Okay, but there's something grow out from that amputation. And of course, the best will be complete regeneration. In this complete regeneration, you just regain that structure. You regain a new limb that, no matter morphologically or functionally, that's just like a brand new, perfect one. Okay, so this describes the scale or power of regeneration. And then on top, it shows you in different species how they can achieve different outcomes. Like in mouse, maybe in adult, you don't have any regeneration power. Therefore, the regeneration is limited by healing with a scar. But in embryonic mouse, so if you remove the digit tip, the tip of the digit in embryo, that's before they are born, right? But if you do a macrosurgery, you go in there and you take the tip of the digit, it's going to regenerate back. This sounds like so trivial, right? But that means the animal at least have some regenerative power. As you can see, the regeneration is around here. It's scar-free, and it should be hypomorphic. That means the regeneration is not complete. It's just you gained a little bit of a digit tip, not completely. Now look at xenopods. In late stage xenopods, that means the adult xenopods, and you can only have scar-free wound healing if you remove the limb. it won't give you back the limb. But in early stage, like when they're young, when the amphibian is young, then you can have complete regeneration. Only in young individuals, not older ones, even for amphibian. So what we're going to discuss today, if the scientists are doing experiment, it's always in very, very young individuals, not in old ones. And here I show the two scientists who are best known for their work in limb regeneration. The one on the left is Jeremy Brooks, and the one on the right is Eli Tanaka. The Eli Tanaka is a student, I would say it's a trainee of Jeremy Brooks, and they both contribute to our understanding of limb regeneration. And probably it was mentioned that Ellie was a biochem student, graduated from Harvard, and then she went on to study her PhD somewhere here. I couldn't remember her institution. And then she moved to UK. That's University College London. He worked with Jeremy Brooks, and then she learned the strategy to use the amphibian as models, but both contributed. I would say I would say maybe the majority of our understanding of limb regeneration. And Jeremy mostly used Newt. That's the creature I showed at the very beginning of the previous lecture. The golden one is called a Newt, or sometimes called Urodela. Urodela. Urodela is the other name for Newt. They're amphibians very closely related to each other. All these creatures are closely related, but they have different names. So Eli Tanaka used the Mexican salamander. As shown here, it's also named axolotl. Axolotl, this one. Okay, that's the model system she used. So I will introduce an important concept in regeneration. So this concept is called blastima. I didn't describe blastima in flatworm, but in flatworm, when you cut the animal to several pieces, the blastima will also occur. So what is a blastima? So basically, when you do an amputation, it's a very clean cut, right? It's a very sharp cut there. But at the first stage of regeneration, what you're going to see, of course, is not a regaining of the structure immediately, of the limb structure immediately. They won't, of course, it won't occur like that. But instead, you're going to have this tumor-like structure. So basically, you have a sharp amputation, but then quickly, like within several days, you have this thing called a blastima, as shown here. So this line is where the amputation was done. So there was a sharp edge there, but quickly, within days, the animal is going to build this structure called blastima there. So this happens in flatworm as well. So at the beginning, when you cut them to pieces, at the end of those cuts, you're going to have this kind of structure called blastima. The blastima, of course, is covered by epithelium. So it's covered by epithelium. Basically, the wound is now covered completely by the growth of the adjacent epithelium, right? Just like if you have a small cut on your arm, it gets covered by epithelium. But something's still going on inside. Although the epithelium will cover the surface, so bacteria or any other things will not be able to enter. But within that structure, things are happening. So as you can see, you see a lot of cells when you compare it to the left side. The left side is the remaining part of that stump, right? But in this newly created part, this blastima, there are a lot of cell growth. Tons of cells when you compare the left to the right. All of a sudden, you have proliferation, a lot of cell proliferation there. So you will learn later on that the regeneration is guided by the epithelium here and the proliferative cell, the blastema cells. The cells here are called blastema cells. So these blastema cells are the most important part for regeneration. So these are the progenitor cells. The blastema cells are the progenitor cells that proliferate before undergoing differentiation and take their fate, because later on they will have to become muscle cells, epithelial cells, blood vessels, all kinds of cells needed for the new structure, right? Whatever is in that limb, that will have to be regenerated by the blastema cells now at the early stage of the regenerative process. So blastema cells are amazing. So they have a remarkable property that kind of retain the positional identity. So here is one example here. So let's look at B, the B part of that amputation, the B part of the experiment. So basically, the scientists did a proximal amputation. Proximal means upper arm. For us, it's upper arm, right? It's for this limb, it's the upper limb. They did the amputation here, and then, of course, the animal will generate a blastema. That's a growth of a new growth here, right? Not get the wound covered, and some cells are proliferating here. Now, they cut this blastema part away. So after the animal generated this blastema, they cut this away. Okay, now they did another amputation on another animal, a different animal. But this amputation is done at the distal end. So it is the further part of the limb. Okay, so the distal part of the limb, they cut that. Of course, at that stage, you don't have a blastema yet for this new amputation, because the blastema will take time to form. Now, what they did is put this blastema on top of that stump. Okay, so you transfer the blastema from the upper arm toward lower arm or lower limb. So guess what will happen? Amazingly, this blastema is going to regenerate almost the full length of that limb. The reason is because this blastema was cut from, this blastema was formed because of upper limb amputation. So this blastema have all the information, positional information there. So when they are put to the lower limb amputation, they don't care. They just regrow the full length limb there. And similarly, like I put in text there. So if you do identical cut here, so you're going to form two blastema, right? So if you do, so in one individual you do this upper arm amputation, in another animal you do the same upper arm amputation. Now, after they form this blastema, you remove one blastema, you put on top of that of another blastema. Amazingly, that animal is going to generate two limbs, full-length limbs. Again, showing you that the blastema itself have all the information as to what to become. Okay, ask please.
So the blastema. So the thing is that it can't grow on its own, right? It needs to be connected to somehow the body in some way, correct?
Certainly.
Okay.
Yeah, it has to be put on top of that stump.
Yeah,
it cannot grow by itself. So in one simple explanation is it won't have the nutrient to get support, right? Yeah. Another thing, I'm glad you asked a great question. So another thing, the neuron plays a role. So the neuron from the remaining part, the stump, plays a role as well. So if you destroy the neuron from the remaining part, the regeneration cannot occur.
Interesting.
Yeah, it's not only blastema, of course.
Yeah, but it could be
Okay.
the factors.
Yeah,
Yeah,
but
gotcha.
here is at least it shows the blastema have the positional memory.
Yeah.
Yeah.
Okay.
Okay. So here I want you to compare the blastema or the cover the cover of the blastema to embryonic development. So I want to explain to you why this the cover, the epithelium cover, also plays an important role. So here, look at my mouse I'm pointing to as the distal end of a growing limb bud. Oh, I'm sorry, it's not that part. In this blastema, we say this epithelium, newly formed epithelium, originally you don't have the epithelium. It's because of the growth of the remaining part, right? You have this epithelium there. And this is called the apical ectodermal cap. It's called apical ectodermal cap, or AEC. This part is called AEC. So this AEC is sort of comparable to an embryonic structure called apical ectodermal ridge. The one called ridge, the other is called cap. This is called cap because it covers the stump, the amputated region. You have a stump there. So the AEC, or apical ectodermal cap, is the cover. But during embryonic development, maybe this is a human or mouse, I cannot tell, but there is structure called limb bud. At early developmental stage, the arm or your leg is actually a bud here, as a bud here. So this is the bigger box is the amplified view of the limb bud. But in this limb bud, the limb bud has, the cells have already gained positional memory already. I shouldn't say memory because at this point it's not developed yet. They have collected positional information in the cells already. For example, the limb bud can be described as anterior, posterior, proximal, and distal. But later on, when they develop into the adult structure, you have to obey the information originally described in the limb bud, like the anterior, posterior, proximal, and distal information. Basically, which part from the distal will give rise to this hand is already encoded even at the limb bud stage. So why I mention this is because you can do a comparison in this blastema structure covered by AEC, the apical ectodermal cap. It's quite comparable to the limb bud. They both have the positional information, so they know maybe the top part will give rise to the little finger, while the bottom part will give you the thumb, not the other way around. So they know the detailed information of their position, even at the stage of blastema.
Professor,
Yes.
what's the level of potency for blastema cells?
So we're going to describe that right here. So in the next slide. Okay. Here, the scientists try to understand this question. So let's think about potency of the blastema cells. A view, a straightforward view or opinion from us could be like maybe all the blastema cells are adult stem cells. They are multipotent, right? And now they can differentiate toward the many cell types that is involved in the development of that limb. For example, like epithelium, muscles, bones, vessels, right? You would think the blastema cells are multipotent, right? They should give rise to all the different cells. That's our idea. If we have looked at the result from the previous slide, we think probably that's the case because there are multipotent stem cells. Now, you have to prove that. So Ali Tanaka did experiment. What they did is pay attention to A here, please. So again, it's about transplantation. So they did a transplantation experiment. So they make one animal to be a transgenic animal. Therefore, the animal will have green bones. So the bones are green, right? Because some genes in the bone now carries a reporter, like we described many times. If you put a GFP under the control of a gene, that gene is specific for bone. Now you can make the bone to become green. That's what they did for one animal. And then now they take a small piece of that animal, of this donor animal, and then put this small piece into a recipient. But this recipient doesn't have a green bone. Okay, it's just a regular bone without the indicator. So if you put this small piece of bone to the recipient, only this small piece will be green fluorescence, positive, right? And next, they will do amputation. So around that transplant, they will do amputation. Now they will study the blastema and the regeneration. So that's the idea here. So because this blastema apparently will have something to do with the green bone, right? The transplanted bone, because that's where you do the amputation. So the blastema will have something to do with that newly transplanted bone there. So now let's observe what will happen. So first thing they know, when you do the transplantation, when you put that green bone to the recipient, it's not only bone. Although you did just bone, you try to do a clean experiment, but it has carryover. For example, like some muscles get carried to the recipient animal. You can't avoid that. No matter how you did the experiment in a clean way, you can't avoid carryover of other cells. So that's first the thing they observed. You have carryover. And then after amputation, of course, you see the green fluorescence because that's the remaining bone there. And now you see the bone now provides some cells into the blastema because the white line is where the amputation occurred. That's the clean cut. And then the part of blastema is there. Some green cells contribute to the blastema. And then later on, when they see they observe the regeneration, they found that that bone only contribute to the new bone. Okay, so the transplanted bone didn't contribute to other cell types in the new structure, only contribute to the bone. It's known that the bone won't grow into the new structure. It's because of the blastema. So that the information you get from here is the bone cell will give rise to some blastema cells, a subset of the blastema cells. Now the bone cells become progenitors, bone progenitors only, because eventually this bone cell will, the blastema cells that have a bone identity eventually will give rise to the new bone in the new structure, not muscles. And here is a summary of that finding, because they not only did bone, they did many other structures as well. They found that the epidermis cell will give rise to blastema cells that bears a mark of epidermis. Eventually, those blastema cells will become epidermis only. The Schwann cells is a neuron cell. They will give rise to Schwann cells only. The muscle cell will give rise to blastema cells with muscle marks, and then eventually become muscle. The only slight nuance here is the skeletal muscle. So the skeletal muscle, of course, give rise to skeleton, as shown here. But sometimes the dermis can give rise to both dermis and skeleton. So the dermis that can become a blastema cell that have some kind of multipotency. Only for the dermis cells that have multipotency, now they can become dermis and skeleton. That's what their conclusion here. That's amazing. That's big. If the scientists that didn't do this experiment, didn't publish this work, we would think the blastema cells are multipotent, but actually they're not. They're unipotent, mostly. And the blastema cells are derived from the remaining structure. The remaining structure somehow has the power, has the positional memory. Okay, this is, yes.
So that I think like the blastema consists of a lot of different cells. They can regenerate themselves, but not like homogeneous. It is heterogeneous. Is it correct?
Yes, it is correct. So the blastema is a mixture of many different cells, although they are all called blastema cells, but they have different tissue origin.
Yes.
Yeah, the bone will give to some blastema cells that have a mark, epigenetic mark of the bone identity, and also the positional identity.
Yeah.
As we learned earlier, if you cut from the upper limb or lower limb, the blastema cells are different. So they carry the cell specificity information as well as positional information.
I see.
It's quite amazing, you know. That's why this work is a scientists give a lot of great comment about this discovery. So additionally, what they have shown is, again, this is basically what I said in the previous in my explanation to the student who just asked the question. So basically, they not only carry now, like I said from the beginning, the blastema cells will have positional effect, like shown here. The blastema cell have positional effect, and then in the new slide, as shown, I'm sorry, on this slide, the information is it's not the blastema cell that carries the positional effect. It's because of the remaining part. Okay, let's observe this experiment. So basically, it's a similar transplantation. You remove a piece of the donor, which is green, you put into this recipient, but now you do an amputation right in the middle of that transplanted green bone. You do that, and of course, as a control experiment, you put the distal part into the same region, right? In the distal part, you put into the same region, although this amputation is the transplant in the recipient is in the upper level, but of course you can choose to put the same bone piece or a different bone piece. Therefore, in the some of the experiment, you just take the distal bone from the donor and then put to the same place. So now let's observe what will happen after amputation. So basically what the scientist has shown is if you have the same bone put into this upper arm level, you do amputation, it's going to grow a full arm, okay, as expected, as we have explained. And now, if you put just a small piece of bone from the distal part you put into the upper arm, you still are going to grow a full arm, but only the distal part of that structure is contributed by the green cells. So what does that mean? That means the remaining part carries the positional information. Therefore, if the transplant is from the distal part, during regeneration, you can only regenerate the distal part out of the distal implant. But why? Your question is why the whole structure can be regenerated. That is because when you do the amputation, it's not only about bone, it's about all the other tissues as well. So that's why you still grow a full limb, but you know the in terms of bone contribution, only that part, the implant part can only contribute to the distal side of the bone, not the upper bones. So this again demonstrates that the information is carried by the remaining part, and again demonstrated, it's not the blastema, but the remaining tissue that contribute to blastema that carries the identity information as well as positional information. Now, I guess the important question here is two questions. One, how the position effect is memorized. So I guess it's different from flatworm because now the cells may be, because we demonstrated, the scientists have demonstrated that the remaining part, the remaining tissue carries the positional effect. So it must be both the cells and probably the environment as well. So the cells, in the upper arm and lower arm, although they're both bone cells, but their epigenetic information are different. Epigenetics are different in those two cell types. Although they're very similar, but the position are somehow kept in the remaining cells. So that's one explanation. Please distinguish that from what I explained for platform. Platform is more about the environment, but here the cells are involved. Another important question is, how do you know that stem cells are not involved? Maybe the blastema cells are stem cells. They are not progenitors. They are stem cells. So if they're stem cells, you would think, okay, there is a de-differentiation from the remaining tissue, and now it has to be stem cell first and then differentiate. So here, the scientists did some experiment. So they basically use CRISPR, the technology I described in multiple experiment. So the PAX7 is negative for the somatic cells, but it's only positive for stem cells. For stem cells, you're going to have PAX7 positivity. So if you link PAX7 to a GFP marker, and you can observe if you have involvement of cells that express PAX7 during the process. But in the experiment, at least in their experiment with, let me see what is that. Okay, so in this process, in this experiment, they don't see positivity of PAX7 cells. That means the cells only proliferate and give rise to some progenitors, but they never go through a stem cell stage. Because if the cells become stem cells, that PAX7 GFP will be able to indicate the presence of them. But the answer is no. Okay, therefore, in this particular experiment, they don't have the involvement of stem cells. Okay, so this is the very last slide. I guess I will skip it. Basically, it tells you that in the zebrafish is another model that people use for studying regeneration, and just like this is not tetrapod, it's just a fish, right? But it's vertebrate. It shows if you cut the apex, that's the very end of the heart. If you cut that in young animals, they can regain that part of the heart. But if you cut a little bit higher, they can't. So that means, for the internal organs, the regenerative ability is now much less retained. Group 5 is going to present a paper this Friday, and the paper is exactly the same paper that I described. I hope Group 5, if you're here, please pay attention to describing the markers such as PAX7, MIF5. When those markers are positive, remember to explain to the class what's the meaning of that. Okay, so yes, I didn't go into the details there.
Professor, so we still represent what you presenting and then go deeper into the marker. Is that what you want?
Yes. So right. I want to, when in figures, when you see something like a MIF5 positive, yeah, PAX7 positive, remember to explain to the class what is MIF5. Yeah. So when MIF5 is positive, what's the meaning of it? Yeah. Instead of just say this is MIF5 plus positive. Okay. Yes, yeah, explain that. Okay. one more thing. I have a doctor's appointment at 9. There is a slight chance I may not be on time to our class, but please go ahead and present. But remember to record your presentation so I will know. If I miss some part, I will still be able to judge your score, give you the score. Okay.
So do we need to generate the meeting out on or at the end a meeting that we occurs?
If you don't see me,
All right.
don't delay. Just get your presentation started, and then, but keep a video record.
All right. So it's supposed to be a meeting, even though you are not. Like,
even if I were not here, because I have a doctor's appointment at 9. I think I will be able to come back, but there is a slight chance.
Yes, sir.
Okay. All right. Sorry for being out of our time again. So if you have a question, you can stay and ask me. Otherwise, feel free to leave.
I have a question.
Sure.
So when you amputate the limb, the blastema will regenerate the whole limb back. When you take a small piece of, let's say the bone, it just regenerates the bone. It doesn't regenerate the whole limb. That's what I understood. Is that right?
Not true. If we're talking about the very last experiment, okay, here, remember, try to understand the process here. So they have one animal that's green fluorescent positive. So that's the donor animal. Then they put some piece of the bone from the donor animal to a recipient. So in some of the experiment, what they did is exactly the same bone they put into the recipient. Like you can remove this part here, right? And then you take away the counterpart from the recipient and you just put the same bone there, right? This is in one experiment you can do this way. But in another experiment, what you can do is you remove this piece from the recipient, but what you put in here is not the counterpart from the donor, but it is the distal fingertip put into this same region. Okay, this is two scenarios. One is put the exactly same bone to this upper limb region. One is put the fingertip bone now to the same region. So you with me?
Yeah.
Okay. And now next, what you did is amputation. So you did the amputation. Now remember, when you do the amputation, it is on different bones. For the two comparisons on different bones. So you do this amputation. The top is when you have the same piece put into the same place. So of course, the green bone is going to contribute to the bones in the new structure everywhere, not only the upper part of the bone, but also the lower part of the bone everywhere. So that shows that the bone here have positional effect of everything through this structure. But in the second experiment, what you did is what you put in there is the fingertip. Okay, what you put in there is only the fingertip, not exactly the fingertip. It's just a part of the finger. Now, when you do amputation, the same thing happens. You have blastema, but look at the green fluorescence. The green fluorescence is only around the digits, right? It's only around the digit, not in the upper arm. Why? It is because for this bone here, located at the end of the structure, it only has positional memory of the fingers. Therefore, it can only contribute to the bone of the fingers. It cannot contribute to the structure in the upper part.
Makes sense now.
Okay, so the upper part is from something else, like here.
Do you mind going back to the previous slide? Yeah, it's just like I think you said that when you amputate the limb, you get the whole limb back sometimes. But if you take a small piece of it, you just get that piece of it because of the positional effect, right? That's what I'm trying to.
Not exactly. So in this experiment, what scientists is trying to determine is individual tissue. You probably get confused because when you do amputation, the growth, the regeneration is not only about bone. It's about bone, muscle, epithelium, all the cells there. So when you do an amputation, the regeneration usually will be complete, regardless of the transplanted bone or not. The regeneration itself will be complete, no matter what. So what the scientists are trying to do is observe what this green part, that's the part that they can control, they put into the animal, they study what this green part will become. They're trying to understand the details, the mechanisms, but that regeneration itself is going to be complete anyways.
Yes, so they're amputating the whole limb. They're not just taking a piece of just like the bone, for example, right? They're amputating the whole limb.
Yeah, that they get everything removed. Basically, you cut right here, right? And everything in the distal will be gone.
Yeah, I just thought they were taking the green part only.
No, they're when they cut. So you remember this experiment is two part. One, the first is a transplantation of a bone piece. That's the first part of the experiment or preparation. You
Okay.
have to transplant that bone piece to a new individual, and then in that new individual you perform amputation. Now you can study the green bone, which is under your control. It can show green fluorescence. Now you can observe what that green bone will go through during the process. But the whole process will involve a lot of things, not just that piece of bone.
Okay, makes sense now. Thank you.
You're welcome.
Professor, I have a question about the planetria that treat with radiation.
Okay.
So I have read some papers that irradiation of different cell gonna somehow give the remaining cell a power to have more power for regenerate or for healing. Have you
The
heard that?
the the in this irradiation, if it is not the extremely high dose, okay, if the dose is not extremely high, it kills. It won't kill all the cell.
Yes.
Leave some cell behind. But if it is extremely high, it can kill all the cells in there, and therefore the one you can study if one cell will be able to regenerate. But like you said, the cells is the source of the growth factors I was talking about.
Yes, but I just read like somehow the radiation treatment itself, like the radiation have something that have the remaining cell become like more potency or have more power to help another cell. So it should, I don't know, I just have read some paper about it because our paper also they use like irradiation rescue
Mhm.
for the squamous cell. So there are like previous paper they said that with the radiation treatment,
Mhm.
somehow it helped the remaining cell have some more power compared to normal.
That's interesting. I
Yeah.
didn't know that. Yeah, let me read those papers. Like I said, now this is a new field actually. The using
Oh,
this as a model system is a new field. As I said before, I teach this class, I didn't even know the existence of this. This
oh.
is very powerful, right? So
Yes.
I didn't know that the irradiation can make the remaining cells to get changed somehow.
So in our
Mhm.
paper, they didn't mention about it, but they just use it, and they said that the irradiation didn't have an effect on their experiment. But I
Mhm.
just Google back like why they did it, instead of just amputation, they do the radiation to amputate it. And
That-
then there's some paper about irradiation treatment or something like that.
Okay. That's interesting. Basically, you can tell that when you do amputation, you don't have the growth factor effect will be much less important if it is an amputation. If it is irradiation, now you probably can study the environment, how the environment triggers different cell fate, because you still pretty much maintain the structure if you just do irradiation. It's not going to kill all the cells. The stem cells are more sensitive, so you can study how the environment affected the stem cells to take different fate if you do irradiation. But if it is amputation, then I will feel that the model will become more towards the information kept in stem cells, right? Because now the structure is damaged. If you do amputation, the structure is damaged. It's not
Yes.
like when you do a irradiation. Yep.
I see it. Thank you, Professor.
You're welcome.